Kia ora. Welcome to day 9 of the COVID-19 Masterclass. I'm Jackie Haddingham, manager of the Otago Global Health Institute, OGHI for short. Today's webinar will run for an hour. We'll finish at around 1 p.m. New Zealand time. Our webinars are being recorded and will be available on the OGHI website later. We're also live streaming through the OGHI Facebook page. During the webinars, each speaker will do their presentation, and you can ask questions at any time through the Q&A function at the bottom of your screen. I will then read the questions aloud for the speaker to respond. If we're not able to get through all the questions during this session, we'll ask the speakers to provide written responses, which we'll post on the OGHI website as well. Our first speaker today, Professor Miguel Quiñones Mateu, holds the Webster Family Chair in Viral Pathogenesis in the Department of Microbiology and Immunology at the University of Otago. His talk will reveal what has been going on in the Otago COVID-19 laboratory. Miguel.
Thank you, Jackie, and thank you for the invitation, and thank you to the Otago Global Health Institute for, inviting me to talk about what we have been doing in our lab. I'm going to tell you a little bit about the history of why we started working with these virus. You may know that every single virologist in the world became a coronavirologist around February or March. So a lot of people have been asking me what we have been doing for the last few months. I'm going to give you a little bit of a history here. So before we start talking about what we're doing in the lab, I'm going to give you an update. I'm sure that other people have done this, but I'm going to start with this slide. We have more than 10 million people already infected. I will multiply that by 10, assuming that there is a lot of people that have not been tested, maybe more than that. And unfortunately, we have more than 500,000 people that have died because of these virus around the globe. As you know, here in New Zealand, we're doing relatively, really well. We have managed to, I'm not sure if this is going to work. Yeah, it works. We managed to flatten the curve, and we don't have that many more cases, a few important ones lately. But I mean, a relatively small number compared with other countries, like, for example, the US, where we have more than 2.5 million people infected and close to 130,000 people who have died so far. So in New Zealand, based, the government response and the amazing job that everybody has been doing, we're doing relatively well, like I said before. So how this started, and I labeled this the beginning. Believe it or not, I was in Fiji on vacation with my family. I mean, my daughter and my wife, and I started reading the news about what was going on in China with this new virus. And I remember telling them, okay, that didn't look good. I mean, this sounded more like an influenza pandemic again, or maybe even a coronavirus like the SARS in 2003. So I came back to New Zealand. James, who was, you're going to be hearing about, vaccines later that he's going to be speaking, but James came to my lab, to my office in mid-January, and he asked me if I was aware of what's going on in China. And he suggested that we should be ready, that we should be putting together, trying to implement and eventually validate a quantitative RT-PCR test to diagnose COVID-19, at least here in Dunedin. So I said, why not? I mean, to be honest, I hesitated a little bit. I had a bunch of different projects going on in lab, but I thought that this was really exciting. And one of the main reasons that I came to New Zealand to work with emergent viruses, so I said, why not? Let's start working on that. So the first thing that we did, and I'm not going to try to go this really quick because I know that James talked about it last week in this masterclass. So, but I'm going to give you a little bit more of details, technical details. So the first thing that we did was we didn't want to reinvent the wheel. We just wanted to see what other people were doing out there. And this is a relatively simple assay. So we found a group in Germany that had published what it seems to be a really specific and sensitive test to diagnose SARS-CoV-2 infections. So we ordered the primers, and the problem that we had at the time is that we didn't have positive control. Of course, we didn't have any cases here in New Zealand at that point, not even in Australia, I think. So we had to order plasmid and synthetic genes to have it as a control to validate or to test our assay to validate it eventually. So this is around the end of the last week of January that we did this. We ordered the primers and everything, and we started the implementation and verification of the assay in my lab in the Department of Microbiology and Immunology. And this was work done by Blair Lowley in my lab. Just simple, a slide just to show you, because this summarizes a lot of work that Blair did in my lab. The assay was, really nice limit of detection sensitivity. We were able to detect all the way down to 50 to 100 copies per reaction or per ml. Then at that point, we were ready to transfer it to James' team. And Dr. Jenny Grant in the leading hospital was able then to take the assay and implement it in the Panzer Hologics instrument that Blair James described last week. Tested sensitivity and specificity was working really, really well. We tested reproducibility in both labs, and both in the hospital, and of course the assay was really reproducible, and we were getting pretty much the same data both in the Panzer Fusion in the hospital and in my lab in the Department of Microbiology. So at that point, we were ready to transfer it to go live, like James mentioned last week. And then on March 13, James' team was able to identify the first case in Dunedin, which, if I remember correctly, was the 8th case in New Zealand. Okay, they continued working. They did some validation, sending samples to other clinical labs, and everything was working really well, 100% concordance. So we were really excited about that, and continue working. And they continue working, testing samples. This is a snapshot of as of 10 of April, and 10 of April is because of the work that we continue doing. You'll see it in a second. But at that point, we have roughly 1,000 cases of COVID-19 in New Zealand, and roughly 200 cases in the southern districts here in tested here in Dunedin. Okay, this slide is just to show you the demographics of the clinical demographics of the patients that were tested at the time. A little more than 4,600 samples tested. Roughly 60% of them were female, and 190 that were positive as of the 10th of April, like I said, and roughly 50-50 female, male at that point. This is the distribution by age of the number of people tested positive. And I don't pay too much attention to this. We need to investigate a little bit this more, but this could be just resemble the demographics of the Dunedin, or at least of the southern, with plenty of young people, students, and a little bit older people like me around the city. So one of the beauties that James mentioned last week about the Panzer Fusion is that it's able to do everything from RNA extraction and to not only diagnose SARS coronavirus 2 infections, but other respiratory infections. So this slide, at that point, his team was able to test up to 335 patients at the time, or samples at the time. The good news at that point was that none of the ones that were cases that were positive for COVID-19 were positive for any other virus or respiratory virus, as tested by the Panzer Fusion. The ones that were negative, 54 patients that were negative for SARS coronavirus 2, some of them were positive for other respiratory viruses, like influenza A was most of them, 37. And many cases were not positive for either SARS coronavirus 2 or other respiratory viruses as well. So at that point, everything was going okay with the diagnostics. They were really, really busy in the hospital, like James mentioned last week. So, but we have this first case in early March. The first thing that I decided to do, or we decided to do in my lab, was that we needed to sequence this. We knew that was positive by PCR, but we needed to sequence and be sure that, well, we had just SARS coronavirus circulating around here. So with the help of Joe Stanton in the Department of Anatomy here in the University of Otago and Jackson Trees, we were able to, basically a little bit more than one day, got the sample from the patient and sequence it using the medium off-core nanopore system. So this is just to show you that of course, we sequenced the full-length genome based on the analysis, the bioinformatic analysis, and showed it seems to be SARS coronavirus 2. But then we took, we just wanted to corroborate that. And we took that first case and that second one that was identified the next day or 2 days later, and did another approach, a different approach. We sequenced against the full genome using a metagenomics approach, this time with the MI6, and took just less than 3 days, to be honest. to sequence the full genome of the both cases. We did identify, as you can see here in this map plot, a few other, bacterial infections, of course, because this is a swab from the respiratory tract that is common, but no other virus. Everything was SARS coronavirus 2. So we are really excited. The PCR work, and of course, the sequencing work, and at that point, of course, we did some phylogenetic analysis, and of course, it clustered with the SARS coronavirus 2, which is, of course, really related to bad SARS-like coronaviruses that are circulating out there. So at that point, actually, even a couple of weeks before that, I made a decision that we needed to work with these viruses. And at that point, I think that we were at level 2 or almost level 3. Many universities were closing, many labs were closing and pretty much going home, like the government was suggested. But I really believe that we really needed to work with these viruses. So with the support of the university, starting all the way from the vice chancellor, pro vice chancellor, dean, and the HOD of my department, we decided that, yes, we should keep these lab open and we should continue working on SARS coronavirus 2. So we were the only lab at some point for the first couple of months that were working in the entire country, and definitely because we had access to our PC3 or physical containment 3 laboratory that is able to work with SARS at the roof of our building. So we continue working on that. We got, of course, all the permits, and it was a hard process to get all this done. But basically 10 days later, we got that first sample that was sequenced, a couple of weeks before. And Rodri has been the one champion. This is Rodri Harford working in my lab, working really hard for the last 3 months, 4 months in the PC3. So taking these pharyngeal swabs from these patients, we basically infected vero cells. We exposed those samples into vero cells, and we were able to isolate 7 different SARS coronavirus isolates. And this is an example. This is Rodri working in the PC3 and cells infected and non-infected with the SARS coronavirus. So I'm going to just take a break here, because at that point, because of the sequence, we really believe that was SARS coronavirus. Based on these, we really believe that was an infection, a viral infection. But we didn't know if the virus that we were isolating in the PC3 was for sure a SARS coronavirus 2 strain. Okay, so what do we do next? Of course, we needed to sequence that. So the first thing that we did was sequence all 7 patient-derived samples. And again, just similar to what we did before, all of them were SARS coronavirus 2. And then the next thing that we did was sequence the virus isolate, the one that Rodri was able to get in the lab. And again, this is the full genome. I mean, not to our surprise, I mean, this is the kind of result that we were looking for. Not only we determined that yes, those viruses that were growing in the PC3 lab were SARS coronavirus 2, but as you can see here in this phylogenetic tree, every single virus matched the sample or the sequence of the virus that come from the patient, so to 100%. So at that point, we were really glad that we have the virus, we have all the sequence, and we were ready to go and for continue working with these virus. I'm not sure if anybody has talked before about the biology of this virus. I'm going to take the opportunity to a couple of slides just to show you. This is the SARS coronavirus. It's an envelope virus. It's an RNA positive strand genome, roughly 30,000 nucleotides. And I just want to highlight these protein, the spike glycoprotein, and everybody has been talking about the spike or surface glycoprotein or the S protein. This is one of the most important, everything is important, of course, in the virus, but this is one of the most important because that's the one responsible for entering the binding to the receptor and entering the cell. And it is one of the main targets for neutralization and of course, for vaccine development as well. Okay. So just like us, everybody around the globe has been sequencing full-length genomes of these viruses. As of yesterday, I think if I remember correctly, there was more than 55,000 genomes of these viruses has been sequenced. And in this database, you can see that people have been tracking all these different, what it seems to be clade. And we can talk about these, and I'm sure that people will have questions about that could or not have phenotypic differences of these viruses that could increase the virulence of transmissibility. So, but these clades or these distribution clusters in the phylogenetic tree are based on, a few mutations that you can see. This is the genome of the virus and the diversity of all these strains. So what we did, of course, because we had the full sequence of our viruses, where these are the different mutations. As you can see, compared with any other RNA virus like HIV or influenza or even hepatitis C, there are not that many mutations. But I want to just draw your attention to these ones, the D614G and the spike, that some people have been trying to associate these with a better replication capacity, at least in vitro and cell culture, or even increased transmissibility. Some people have been saying, oh, this virus is increasing the virulence, it's going mutating, it's going to get worse. Again, we can talk more about that later. So we found, let me go back for a second, we found that basically half of our viruses have that mutation, and the other half, they don't have it. So we did viral growth kinetics, to see how the virus was replicated in viral cells and in cell line, and we don't see any difference between those two viruses. We're repeating that in the primary cells to see if we see any difference, but in our hands, we're not seeing any difference between those two viruses. Awesome. Another thing that we have been doing is with Dr. Bostina in our department. Again, he's an expert in electron microscopy. And for anybody that wanted to see, this is the virus circulated in New Zealand. And that these are the really red dots, of course, artificial colored by Dr. Bostina. But those are individual viral particles in our cells. So what's next? So one of the first things that we did once we got that virus from the PC3 was increase the production and distribution of SARS coronavirus material, including RNA and non-infectious virus. Because if you remember at the beginning, what I was mentioning is that we didn't have any positive controls when we were developing these. So a lot of people have been asking us to use RNA for diagnosis or any other project that they may be interested across the country. So we have been circulating or sharing these material with a lot of, we have been collaborating with a lot of people that are working, developing diagnostic kits for diagnostic tests for COVID-19, especially with Dr. John Stanton, again, with the point of care test, and some using microRNAs-based diagnostics as well. We just got approval for a project to work on disinfection and reuse of personal protective equipment, which is extremely important, as you can imagine. I mean, we have a scarce number of these PPE, and we need to start reusing it. And that's a project that we're going to start running in collaboration with the University of Auckland, Dr. Yvonne Anderson. We're participating in a few clinical studies as well. I'm not going to list all of them, but there are some examples. Of course, as you can imagine, everybody wants to test something against this virus, and because we have it and we have the capability to work with that in our PC3 lab, we have been testing small molecules or chemicals, peptides, peptoids, natural products, even soluble ACE2 receptors. I cannot talk that much about these, but as you can imagine, everybody around the world is doing something similar. Up to yesterday, there are 284 different projects testing these different drugs, and close to 200 in human trials already. Vaccine development, the same. More than 200 around the globe, 23 of them in clinical trials. And we are involved in a couple of efforts here in New Zealand to develop a vaccine for our country as well. And I'm sure that James will talk a little bit more about that in a second. So here, I mean, a lot of people have been asking me about how is it to work in the PC3. So, I mean, I'm going to ask permission here to just show you a 1-minute video of what it is to work in the PC3. And you will see Rodri, what he has been doing for the last 2 or 3 months. I'm not sure that there is that audio there, but I'm going to let you watch the video. As you can imagine, there is a lot of work that has to be done just to get in and out of that facility, and of course, working in the facility itself. So on that note, I just want to finish with acknowledging our team and the people have been working really hard for the last 3 or 4 months, or I would say 5 months now. Ever since I started, like I say, with a conversation with James back in my office in January. Blair and Rodri, my team, that have been, since lockdown 3, 4, we have been working in, side by side in the lab. My lab manager, Dr. Leonor Hernandez, has great support, growing sales, supporting all the work that we have been doing in the lab. Of course, Joe, Jackson, and Robert for the sequencing, Mignia for the electron microscopy, James and Jenny, partners in crime since the beginning, and of course our Institutional Biological Safety Committee and Megan Coleman with all the support of our work in the PC3, just to be sure that we are doing what we're supposed to be doing in the lab. And the funding support from the University of Otago, MB, and the Western Family Chair in Viral Pathogenesis. So, thank you, and questions?
Thank you, Miguel. 1 or 2 questions coming through. Firstly, the presentation was really informative and amazing. Which small molecule so far have you found to be most effective, and which route will be more effective for delivery of drug or vaccine?
Well, so far we in our lab, we're still working on this is early stages. So, and even if we have something, I cannot disclose it. But most people know that the drug that is working the best so far is rendezvous from Gilead in the US. Is a drug that is blocking the RNA polymerase of the virus, blocks the ability of the virus to replicate, and it's currently delivered, intravenously. But ideally, will be to get something that people can take as a pill. Okay, we just learned, I think it was yesterday, that the US is going to try to sequester that, and they're going to try to pile up rendezvous for the next 3 months. So we're not going to have it available for the rest of the world anytime soon. So we need to continue working on these and trying to find alternatives to rendezvous.
Thank you, Miguel. Just a question from me. Can you comment on the significance of the T-cell mediated response as opposed to the antibody mediated response? I believe there is some...
There is a slight difference. Yeah. But just to be honest, I'm going to pass that question to the immunologist rather to the virologist. Right. So I'm sure that James will be talking more about that in a second, and then you can ask him. Yep.
Thank you. Well, we're waiting for a few more questions to come through. Are there any slides perhaps you'd like to go back to, or any other points you'd like to emphasize in your presentation?
No, I just wanted to show what we have been doing in the lab. I mean, stay tuned, because in the next couple of months, we're going to have plenty of data that we're going to be able to share. I just wanted to, tell the story of what we have been doing for the last, few months. Yeah.
It looked like the PC3s are, quite a mission to get in and out of, and the decontamination and the constant health and safety checks must be quite rigorous.
That's correct. I mean, Rodri or whoever is going to work in the PC3 cannot stay there for more than 4 hours. So you have to get all suited and, be trained to work in the PC3, of course. And then after getting out, you have to go and take a shower and all that. So it's plenty of work, but it's really well equipped and suited for this work. So we're really excited. I'm pleased to have the PC3. Without the PC3, we wouldn't be able to do anything.
How do scientists communicate while in the PC3 lab? Actually,
it's really well equipped, and we have Wi-Fi there, computers, we have an iPad, we have a phone. So Rodri has been communicating with me all the time, calling me via FaceTime or Zoom or anything, okay, showing me data in the iPad and sharing that. That's really key, because as you can imagine, nothing can get out of the PC3. Everything has to be autoclaved unless it's inactivated and disinfected and so on. So but if you're working in the PC3 together, it's not allowed. You're able to communicate and talk even wearing those suits. Yep.
Do you see any evidence of attenuation as you passage the virus?
No, not yet. I mean, these virus, we can inactivate the virus with chemical agents, of course, but attenuation, you'll have to pass it. But we haven't done that yet. I mean, we're passing the virus, but it will take a long time. And we're not seeing any attenuation, to be honest, of the virus circulating around the glove. the virus is really successful at transmitting, so I don't think that it's going to happen anytime soon.
Okay. Do you think viral mutations occurred in New Zealand, or were they imported?
Oh no, the virus, every time that replicates is creating or generating mutation and being selected. It happens all the time. These viruses, RNA viruses, like I said before, similar to HIV and all that. But the difference compared with the other RNA viruses that I was mentioning is that this one has a proof-reading ability, meaning that it can go back, the enzyme that is copying can go back and fix some of those mutations. So it doesn't mutate that much compared with other viruses, which is a good thing.
Mm-hmm. Yeah. Where would you predict the most virus mutation to happen? Spike protein, possibly? And other than the ACE, is there another entry site?
Yes. Believe it or not, I mean, similar to other viruses, the envelope is typically the more viable one because those are the ones targeted by the immune system, and the viruses have to, mutate and select for mutation to escape that immune system or immune response. So if you go back, in that slide I was mentioning, some of the mutations were in the spike, but you can see it all over. The reality is that this virus is not mutating that much. It doesn't need to mutate. It transmits so well that there's no selection pressure right now for the virus to mutate. So it's doing pretty well.
It's doing well, yes.
Yep.
How many other PC3 labs are there in New Zealand?
Oh, that's a good question. There are a couple in Auckland, and I think if I'm aware, or maybe it's definitely one in Auckland, a couple in Wellington. I'm not sure they were Christchurch, isn't it? That's true. Okay, so I would say there are 3 or 4 more.
Mm-hmm. Okay.
Maybe you can correct me if I'm wrong.
Would you be able to comment, or would you be able to expand on the types of international collaboration that you and your team have been involved with during COVID-19?
Yes, it's mainly related to vaccine development. We have been collaborating with teams in the US and Canada and hopefully in Europe in the near future. Mm-hmm. There are some collaboration related to antiviral strategies as well, basically drug discovery and development as well, and characterization. But it's mainly related to vaccine development.
Okay. Comment and a question. Awesome presentation, thank you. The video was remarkable. Would the vaccine be just for relevant to strains in New Zealand or worldwide?
Oh, it will work. It will work worldwide. Like I say, this virus, it doesn't have that many mutations. One of the things that we're doing in the lab is, remember that I mentioned that we have two different, they're really similar, but one that had that mutation in the spike and one that doesn't have that mutation. And because a lot of people are putting a lot of attention into that mutation, we're trying to develop inactivated vaccines using both strains, one with a mutation and one without a mutation. Mm-hmm. But there are different strategies that we're going to be following.
Okay. Can you comment on the possibility of antiviral resistance?
Really, really good question. One of the first things that we do when we're working with antivirals against HIV, hepatitis C, or even influenza, once you detect and you know that a particular compound, small molecule, peptide, or whatever it is, it blocks the replication of the virus, the next thing that you do is to try to select for resistance in vitro, because that's going to just check the mechanism of action of that. So that's something that we should do, and I'm sure that people are doing, but it's a double thought because you don't want to develop the drug-resistant variant that could eventually escape, which of course is not going to happen based on what we have, all the regulations that we have.
Yeah. Great. Well, thank you very much, Professor Quiñones Mateu.